Hello everyone, welcome to biology class. In this class we are going to see 12th standard botany chapter 3 chromosomal basis of inheritance. In this part of video we will learn about crossing over multiple alleles. First, crossing over. Crossing over is nothing but it is a biological process, and out of this biological process new combination of genes are produced by interchanging the corresponding segments. So this interchanging of corresponding segments takes between non-sister chromatids of homologous pair of chromosome. First, what is homologous pair of chromosome? Homologous pair of chromosome are nothing but during the time of meiosis. So one chromosome will be inherited from the father, that is called paternal chromosome. The other one will be inherited from the mother, which is called maternal chromosome. So these two chromosomes, one is paternal from father and one is maternal from mother. So this pair is said to be homologous pair of chromosome. So what is non-sister chromatid? So the chromatids of the same chromosome are called sister chromatids. So these arms of the same chromosome is called as sister chromatids. And similarly, the arms of this chromosome is also referred to as sister chromatids. But here in crossing over, the crossover is going to take place between these two chromosomes, and these two chromosomes are said to be non-sister chromatids. So crossing over is a biological process. Out of the biological process, it is going to produce new combination of genes, but this crossover takes place between the non-sister chromatids of homologous pair of chromosome. The term crossing over was coined by Morgan in 1912. And when this crossing over takes place? So this crossing over will take place during packaging stage of prophase 1 of meiosis. So meiosis, already you have idea about meiosis. Meiosis is a kind of cell division. It will take place in the germinal cells. And germinal cells and meiosis itself, you have two, meiosis 1, meiosis 2. And in meiosis 1 itself, you have various stages: prophase, metaphase, anaphase, telophase. So out of meiosis, during the prophase 1, you have several stages: leptotene, zygotene, packaging, diplotin and diakinesis are the 5 stages of prophase 1. And out of the 5 stages, this crossing over will take place during the packaging stage of prophase 1 of meiosis. This is a very, very important objective. When do crossing over take place? At what stage? The options will be given: leptotene, zygotene, packaging, diplotin and diakinesis, like that. So it takes place during the packaging stage. And as I already told you, usually crossing over will occur in the germinal cell. Germinal cells are nothing but the reproductive cells are sex cells. And during the formation of gametes, that process is called gametogenesis. So since crossing over is going to take place during gametogenesis, this kind of crossing over is also referred to as meiotic or germinal crossing over. And this crossing over has an universal occurrence and has great significance. And rarely crossing over can occur in even somatic cells also during mitosis. So when the crossing over occur in somatic cells during mitosis, then that kind of crossing over is called as somatic or mitotic crossing over. Now let's see in detail about the crossing over in stepwise manner. So the mechanism of crossing over is a precise process. It includes 4 step-by-step stages: synapsis, tetrad formation, crossover, terminalization. The first step is called synapsis. So what is synapsis? The pairing between the two homologous chromosomes. So this is, for example, if you take this as paternal chromosome, this as maternal chromosomes, so these two will come into pairing. Intimate pairing is referred to as synapsis. And when this synapsis takes place, the synapsis will take place during the zygotene stage of prophase 1 of meiosis 1. So the homologous chromosome, they come close to each other and they are aligned side by side, and resulting in a pair of homologous chromosomes called bivalents. And this pairing of the chromosomes is referred to as synapsis or syndesis. And in synapsis itself, there are three types. So, procentric synapsis, pro-terminal synapsis, and random synapsis. First, procentric synapsis. So, procentric is nothing but the pairing of the chromosome starts from the middle of the chromosome. If starts to pair, then it is called centric. Terminal means if it is going to start from the tip of the chromosome is referred to as telomere. From the tip, if it is going to pair, then it is called as pro-terminal. And random synapsis is nothing but pairing may start from anywhere. The next stage in crossing over is tetrad formation. Each homologous chromosome of a bivalent begin to form two identical sister chromatids. So, the chromosome will have two identical sister chromatids and they are held together and thereby forming four chromatids. This stage is referred to as tetrad stage. Next, crossover. So, after the tetrad formation, crossing over occurs, and already we know crossing over will occur in pack-18 stage. So, during this crossover, the non-sister chromatids of homologous pair make a contact at one or more points, and these point of contact between the non-sister chromatid where they are getting attached, that point of contact is referred to as chiasmata, singular chiasma. At chiasma, the cross-shaped or egg-shaped structures are formed, and where breaking and rejoining of the two chromatids occur, and this crossover is a reciprocal exchange of equal and corresponding segment. Okay, whatever the segment that is given from one chromosome, the same number of the chromosome will be carried from the other chromosome as an exchange. That's why it is referred to as mutual exchange of chromosomes. And a recent study reveals that the synapses and chiasma formation are facilitated by a highly organized structure of filament. So, this is the picture which is going to show the synaptonemal complex. So, this is one chromatid, sister chromatid 1, and this is 2, and here there is the another pair, chromatid 3 and 4. So, here it is going to occur between the sister chromatid 2 and 3. So, what happens at between these two, there forms a highly organized structure of filament. Those filamentous structure is referred to as synaptonemal complex. And this kind of synaptonemal complex is absent in some species of male Drosophila. Drosophila is nothing but fruit fly. So, it is absent in case of Drosophila, and hence crossing over will not take place. So, this is the crossing over has taken place. The homologous chromosomes, they are aligned side by side. So, here crossing over has taken place, and the last step is terminalization. That is, after it has got crossed over, the chiasma starts to move towards the terminal end of the chromatids, and this process is referred to as terminalization. And as a result of terminalization, the chromosomes, homologous chromosomes, they completely separate from each other. So, here you can see crossing over has taken place. So, here you can very well observe that crossing over has taken place, and here this chromatid is nothing but a non-recombinant chromatid. So, they are same like the parent, so they are non-recombinant, and here you can notice these two chromatids are recombinant chromatids, that is the crossing over have taken place. Here you can find the red color segment, here you can find the blue color segment. So, crossing over has taken place, so it is referred to as recombinant chromatids. So, the stages of crossing over are synapsis, the second stage is tetrad formation, the third stage is crossover, and the fourth stage is terminalization. So, here this diagram is again explaining the mechanism of crossing over. So, they are during the prophase stage, they are coming close contact, tetrad stage, and chiasma, a site where it gets to cross over, and then crossing over has taken place, and then terminalization, they are going to separate. So, this shows the mechanism of crossing over. Importance of crossing over. Crossing over occurs in all organisms like bacteria, yeast, fungi, higher plants and animals. So, let's see the importance. The same can be asked as significance of crossing over. So, during crossing over, the exchange of segments takes place. So due to this exchange, it leads to new gene combination. And as a result of new gene combination, it plays, that is the crossing over plays an important role in evolution. And studies of crossing over reveal that the genes, the order of the arrangement of the genes in the chromosome, the linear arrangement is revealed by studying this crossing over. And genetic maps are based on the frequency of crossing over. And the crossing over also helps to understand the nature and mechanism of the gene action. And this is also very useful for the plant breeding because new combinations are formed. So it is helpful for plant breeding. The next topic is recombination. The crossing over results in the formation of new combination of character in an organism. So due to the crossing over have taken place, so new combinations have occurred. So these new combinations are referred to as what? Recombinants. And in this, the segments of DNA are broken. So here you can see after the crossing over taken place, the segment of the DNA are broken and it is recombined to produce new combination of alleles, and they are referred to as recombinants. And how to calculate the recombination frequency? What is the formula? The percentage of recombinant progeny in a cross is called as recombination frequency, and it is denoted as RF. And how the RF can be calculated? It can be calculated using the formula total number of recombinants divided by total number of offspring. Here you have to mention into 100, so it is missing here. So total number of recombinants divided by total number of offsprings into 100. So you have to write how to calculate the recombination frequency. For the data, I have taken here the coupling configuration cross. So here the parental type is purple flower long pollen and another parent is red flower round pollen. So when we cross these two parental varieties, you can find the recombinant varieties. So out of this, we are getting the parental progeny purple long and red round. And what are the recombinants we are getting? Purple round and red long. So these are the recombinants. So total number of recombinants. So what is the total number of recombinants here? So it is 6 plus 6 divided by total number of offsprings, which is nothing but 44 plus 6 plus 6 plus 44, which is nothing but 100. So 12 divided by 100 into 100 is equal to 12%. So we have calculated the recombination frequency for this cross as 12%. Similarly, when you are given the total number of recombinants and total number of offsprings, we can calculate the recombination frequency by using the formula. And again I repeat, in this formula recombination frequency, total number of recombinants divided by total number of offsprings into 100 will give you the percentage. The next topic we are going to see is genetic mapping. So genetic mapping is nothing but in the chromosome the genes are arranged in a linear order, and the position of the arrangement of genes on the chromosome, the point of contact of the gene on the chromosome, that location is referred to as locus, plural it is called loci. And the diagrammatic representation, so the diagram which is going to show the position of genes and the related distance between the adjacent genes is called genetic mapping. So I'll show you the genetic mapping. So this is the genetic mapping. So in this it shows the arrangement of genes. For example, gene A is here, gene B, gene C, gene D, like that. It shows the arrangement of the gene on this chromosome, and also it shows the distance between the adjacent gene. So what is the distance between the gene A and B? It is shown here. So this is referred to as gene mapping. So the diagrammatic representation of the position of genes and the related distance between the adjacent genes is called as gene mapping, and it is can be otherwise referred to as linkage map. And this concept was first developed by Morgan's student. So he is a student of Morgan, Alfred H. Sturtevant, in 1913, and this gene mapping will provide the clue where the genes lie on the chromosome. Next is map distance. The unit of distance in a genetic map is called as map unit. One map unit is equivalent to 1% of crossing over. So the distance is referred to as map unit, and one map unit is equal to 1% of crossover. and one map unit is also called as centimorgan. So instead of using one map unit, we can call the unit as centimorgan, and it is represented as small c capital M. And this cm is in honor of T.H. Morgan. So we know genetic mapping was developed by Morgan's student Alfred. So for giving honor to his teacher, so he has used Morgan as the unit for gene mapping. So 100 centimorgan is equal to 1 morgan. And for example, distance between A and B gene, if it is estimated as 3.5 map unit. So how you can tell 3.5 map unit? So 1 map unit is equal to 1 centimorgan. So 3.5 map unit is equal to 3.5 centimorgan. Or when we write in the term of recombination frequency, we can call it as 3.5%, which is other way it can be represented as 3.5% can be represented as 0.035. So this is the recombination frequency between these two genes. And what is the use of this kind of genetic mapping? So this genetic mapping is used to determine the gene order. So what is the position of the gene, and what is its locus, where it is located, and what is the distance between the genes. Everything we can know or determine by using this gene mapping. And also the results of the dihybrid and the trihybrid crosses can also be predicted from this genetic map. And also by studying the genes, the arrangement, and how many genes, we can be able to estimate the genetic complexity of particular organism. The next topic is multiple alleles. So what are multiple alleles? So multiple alleles, a given phenotypic trait of an individual depends on a single pair of gene. And the gene is going to occupy a specific position called locus on the homologous chromosome. This is usual. So gene will occupy a specific position on the chromosome. But when any of the 3 or more allelic form, okay, so usually there will be a single pair of gene too. But when 3 or more allelic form of a gene is going to occupy the same locus in a given pair of homologous chromosome, they are called as multiple alleles. So this is the definition for multiple allele, very, very important. 3 or more allelic form of a gene occupy the same locus in a given pair of homologous chromosome. They are said to be multiple alleles. And there is one check your graphs question. There may be multiple alleles within the population, but individuals have only 2 of those alleles. Why? So there may be multiple alleles, more than 2 alleles in a population of a trait, but a person, an individual, can inherit only 2 of the trait, because he is going to inherit one from the father and one from the mother. That's why the individuals have only 2 of the alleles. Let's see the characteristics of multiple alleles. The multiple alleles will occupy the same locus in the homologous chromosome, and no crossing over occur within the alleles of a series. And multiple alleles are always responsible for the same character. And the wild type alleles of a series exhibit dominant character, whereas the mutant type will influence dominance or an intermediate phenotypic effect. And when any of the mutant alleles, multiple alleles are crossed, the phenotype of the offspring will be of the mutant type and not of the wild type. These are the characteristics of multiple alleles. The next concept we are going to see is about sterility in Nicotiana. So in plants, multiple alleles have been reported in association with the self-sterility or self-incompatibility. What is meant by self-sterility or self-incompatibility? So it is nothing but when the pollen from a plant is unable to germinate on its own stigma. So the pollen of the same flower is going to, if it is not going to germinate on its own stigma, then there will not be any chance of fertilization also. Such condition is called self-sterility or self-incompatibility. Who have observed this self-sterility? It was observed by Edward East in 1925, and it was observed in Nicotiana. And the genes for self-incompatibility in Nicotiana can be designated as S and this S as different allelic series like S1, S2, S3, S4 and S5. So what happens? The cross-fertilizing tobacco plants were not always homozygous as S1, S1 or S2, S2, but all the plants were heterozygous, because if they are of the same type, they are not going to be fertile, they are going to be sterile. So always they will be heterozygous, S1, S2, S3, S4. So it is going to be different, 1, 2, like that. Definitely it is not going to be homozygous, 1, 1. There is no chance, because they are exhibiting self-sterility or self-incompatibility. So when crosses were made between different S1, S1 plants, the pollen tube will not develop normally. So when the cross is made between S1, S1 and S1, S2, S1, S2 and S1, S2, so it is not going to produce a fertile offspring. So it is going to be sterile. But effective pollen tube development was observed. So pollen tube development was observed when the cross is made between S1, S2 and S3, S4. So when crosses were made between the seed parents with S1 and S2 and pollen parents with S2 and S3, two kinds of pollen tubes were distinguished. That is, the pollen will carrying from S2 were not effective, but the pollen carrying S3 were capable of fertilization. But the cross from S1 and S2 and S3, S4, all the pollens were effective. So I'll tell you about the progeny now, and again I'm going back to the previous slide and I'll explain. So here you could see the pollen is designated, the allelic form is designated S3, S4, and here this is the allelic form of the stigma. So what happens when the pollen is having a different allelic form than that of the stigma, the pollen are germinating. But here in this case, when you see the pollen, the same allelic series, the pollen tube will not form, the pollen grain will not germinate. So here also the pollen has got S1, and here also if it has got S1, so the pollen tube will not develop from S1. So the pollen tube can develop only from S3 in this case. Similarly, when you see the other example here, here also S1, S2, and here also the pollen is also S1 and S2. So there is no germination of pollen grain, thereby no pollen tube formation. So all these cases, they are said to be self-sterile or self-incompatible. And here they have given the female parent and the male parent. So when it is going to be same, it is going to be self-sterile. So here is one example, S1, S2, here also S1, S2 self-sterile. And another example, S2, S3, here also S2, S3 self-sterile. And another example, S3, S4, here also S3, S4, it is going to be self-sterile. So the other crosses, S1, S2, here S2, S3, S2, S2, both are same. So this pollen will not germinate. So the chance of the offspring will be S3, S1 and S3, S2. And similarly in the other cases, all the pollen grain can germinate and leading to the formation of 4 kinds of progeny. And the second type, S2, S3 and S1, S2. So here 2 is there. So only S1 can form the pollen. So the offspring will be S1, S2 and S1, S3. So this is going to be self-sterile, already I've told you. And here also, here pollen also S3 is there, and here stigma spot also S3 is there. So the S3 will not germinate. So S4 alone will germinate, and the offspring type will be S4, S2 and S4, S3. And similarly for the other stigma spot and male parent, that is pollen source. Next we will see about the sex determination in case of maize. The binomial name for maize is Z maize. It is an example for monoecious plant, that is the male flower and the female flower are present on the same plant. Under 2 types of inflorescence we can observe. This is the terminal inflorescence. So it is located at the tip of the inflorescence axis, terminal inflorescence, and this terminal inflorescence have staminate florets. And this is referred to as tassel, whereas the lateral, that is the branches, will carry some inflorescence which is referred to as lateral inflorescence, and it will develop the pistillate structures, and it is referred to as ear or cob or silk. And unisexuality in maize occurs through the selective abortion of stamens in ear florets. So how they are producing unisexual flower? So there will be a selective abortion of stamen in case of ear florets and abortion of pistil in case of tassel florets. And a substitution of two single gene pairs, BA for barren plant and TS for tassel seed, makes the difference between monoecious and dioecious. Dioecious, that is, rarely the maize plants will be having separate plants for male and female. And now the explanation of the sex determination in maize. So here you can find the genotype, and which is the dominant genotype which is carrying the superscript plus is said to be the dominant, and all the other are going to be the recessive. When the genotype is BA and TS, that is both are recessive, double recessive, then the sex of the plant will be, it is a rudimentary female, and here there will not be any silk, the female part on the stalk, but transform tassel part will be transformed to pistil. That's why it is a rudimentary female. And in the other case, BA, TS plus, here you have TS plus. So TS stands for tassel plus, and hence it will be a male. And both are dominant, double dominant. So the plant will be having both the male inflorescence tassel and female inflorescence cob. So the sex is monoecious. Both male and female flowers are present in the same plant. And next only BA plus and TS. So this is dominant and this is recessive. And since BA plus is there, it is going to bear the cob. And since TS it is not having the plus, it is going to lack the tassel. So this sex will be normal female. And here this table shows the sex expression based on the combination of the alleles, and most of the mutations are shown to be defects in gibberellin biosynthesis. Gibberellin is a plant growth hormone, and gibberellin play an important role in the suppression of stamens in the florets on the ear. So in the ear floret is nothing but the female inflorescence, and in the female inflorescence the gibberellin play an important role in the suppression of the stamens and thereby producing the female flower. So that's it with today's part of the video. So in this part of the video we have seen crossing over, also we have seen about recombination, we have seen about the genetic mapping, multiple alleles, and self sterility in case of Nicotiana, and sex determination in case of maize. Meet you in the next video. Thank you.